Hello everyone, and welcome to this webinar titled Impact of Depth Filtration of Disulfide Bond Reduction During Downstream Processing of Monoclonal Antibodies from CHO Cell Cultures, hosted by Biopharma Asia magazine and presented by Brian O'Mara, Senior Research Scientist at Bristol-Myers Squibb, and Alexei Voloshin, Global Application Strategy Specialist at 3M Separation and Purification Sciences Division. My name is Stephen Edwards, and I will be your moderator. Now, please allow me to introduce our first presenter. Brian O'Mara is a senior scientist at the Downstream Bioprocess Development Group at Bristol-Myers Squibb in Seattle, Washington. He has been working in the biotech industry since the year 2000, with previous experience including YF with Pfizer, University of Rochester, Genencor International, DuPont. He has worked on the development of a variety of proteins for both clinical and commercial development, including monoclonal antibodies, vaccines, enzymes, protein conjugates, and radio-labeled diagnostics from both microbial and mammalian expression systems. Brian received his BS in biology from Binghamton University and MS in chemistry from Lehigh University. I will now be handing over to our first presenter. Welcome, Brian.
Thank you, Stephen, and thanks to Biopharma Asia for the opportunity to share some of our work that we've had investigated on the depth filtration unit operation and how it relates to antibody reduction. So the title of my presentation today is The Impact of Depth Filtration on Disulfide Bond Reduction During Downstream Processing of Monoclonal Antibodies. A quick overview of presentation here. We'll be discussing some of the observations of antibody reduction that we observed during large-scale production, dive into the root cause analysis, and spend some time discussing the investigation of the depth filtration unit operation as the causative factor for antibody reduction, and then wrap up with the conclusion. So in this schematic here, we've got a process flow diagram for the monoclonal antibody production overview. So the top part of the schematic represents the upstream, and the downstream is represented on the bottom. So with the initiation of the cell culture, cell bank vial is thawed and expanded through a series of seed bioreactors, and ultimately a 2,000-liter single-use production bioreactor is inoculated. After 14 days of production, the material is harvested through depth filtration, and the clarified harvest is stored in single-use bags. Over a period of 4 days, the protein A chromatography column is cycled, one cycle per day, and is subjected to a series of chromatographic and filtration unit operations to ultimately produce final drug substance. When we had performed this purification in GMP production, we observed a low purity in 3 of our protein A cycles, 3 of the 4 of the protein A cycles. The first cycle was maintained at greater than 97% purity by non-reducing CE-SDS, and cycles 2, 3, and 4 all measured less than 1%. So starting on day 2, or cycle 2 of the protein A capture, our purity of the protein A viral inactivated bulk decreased to less than 1%. Analysis of the protein A eluates by size exclusion chromatography and non-reducing CE-SDS showed that the protein A eluate detected here under non-reducing conditions showed a mixture of reduced heavy and light chains in the protein A cycle 2, which measured at less than 1% purity by CE-SDS. On the right-hand side is the size exclusion chromatography profile showing that both reduced and non-reduced protein A eluates migrate as a monomer with very low levels of low molecular weight observed. This is indicating that the antibody is intact in the reduced sample and being held together by non-covalent forces. So those subunit dissociations that we see in the reduced species only are detectable under denaturing or SDS conditions. So going into the second purification under GMP, maintaining that dissolved oxygen in the clarified harvest was discussed previously by Genentech as a risk mitigation strategy to prevent the reduction of the antibody in cell culture. The presence of oxygen or high DO levels inhibit the reduction mechanism and also promote reoxidation of the reduced antibody. In their case, they had done an air sparging technique to maintain that oxidative environment. And in our single-use bags that we had during GMP production, air sparging was not feasible. So to maintain the DO level, we had provided a headspace in the bag of approximately 15 to 20% to accommodate for clean compressed air circulation in the top of the bag. So the picture on the right-hand side displays the air in and air out strategy that we use to maintain the appropriate amount of dissolved oxygen in the clarified harvest tank. So we had done this and held the clarified harvest over a period of 4 days, similar to the first GMP, to maintain the dissolved oxygen levels in the clarified harvest. So as the implementation of the air overlay strategy did resolve our low purity or reduced antibody issue that we were seeing coming off of protein A. So the data shown here in the bottom of the screen showed that the intact MAB was measured greater than 97% in GMP2, where the dissolved oxygen levels were maintained at sufficient levels to prevent antibody reduction. Further downstream from protein A eluate through final UFDF or TFF, we do see differences in how the material further reoxidized throughout the purification train. So the reoxidation of the first GMP, shown in blue, occurred throughout the downstream process, most likely due to increases in pH during transitions in chromatography buffers, and also the passive introduction of air during processing. The purity of GMP2 maintained at high levels of 97% throughout the entire purification train. What's interesting is that the first GMP did not reach the maximum purity after final purification and reoxidation. This is something that we have not fully understood and is currently under investigation to figure out why 100% or greater than 97% oxidation did not occur when the monoclonal antibody began as reduced species. So as our investigation and root cause analysis starts, we've decided to focus in on these 3 unit operations or stages in the upstream cell culture process. So looking at the area highlighted in red here, looking at the production bioreactor, the depth filtration unit operation, and then also the clarified harvest holding conditions prior to commencing purification. So one of the first things we were trying to understand is where this is occurring. So what we were able to do was during GMP2, where we had maintained dissolved oxygen levels in the clarified harvest, we were able to stage some process development conditions where we were able to understand where this is occurring. So as we had discussed earlier, the initial DO was at 100%, and these samples were stored in the GMP lab for up to 3 days with no headspace and no mixing at ambient temperature of approximately 20 degrees Celsius. What we see in the traditional SDS page non-reducing gel on the right-hand side of the screen is that those samples that had been stored for 0 or 1 day had intact MAB purity with low levels or 0 levels of antibody reduction observed. Samples that were stored for 2 to 3 days showed antibody reduction as shown in the fragments observed in the lower parts of the gel. Interestingly, the dissolved oxygen levels in those samples that were stored for 2 and 3 days measured less than 5%, and this is essentially 0% in there. So that's indicating that oxygen is being actively consumed in the clarified harvest during storage. These samples were then re-aerated for 1 hour, just exposure to normal air, and the DOs were restored to 100%, and the samples were rerun on the gel. So what we see is that the samples for day 0 and day 1 maintain the same level of banding pattern in the gel on the right-hand side, and we do promote reoxidation of those samples that on day 2 and 3 that were less than 5% DO. So this is showing that the oxygen is being consumed in the clarified harvest, and also the reduced species are observed in the extended hold times. Also, the reintroduction of air can promote that reoxidation of the antibody once after it has been reduced. So understanding where this is happening in the clarified harvest, the other thing is why. So bioreactor samples from the same GMP were also used to investigate the different recovery methods at lab scale. So the two recovery methods used were batch centrifugation, representing a very gentle or innocuous way of recovering, and then homogenization being the extreme where we get 100% cell lysis. Both of these conditions were centrifuged and clarified, and the supernatants from each condition or recovery method were stored at 4 degrees Celsius with no headspace for up to 4 days. What we see in the picture on the bottom of the graph and the bottom of the slides here is that the centrifuge sample maintained high purity over the storage period of 4 days with no headspace. The homogenized sample, which represents complete cell lysis, resulted in complete reduction of MAB, and that reduction is observed on day 1 analysis. We do see a level or low level of reoxidation in the samples as a function of storage time, and this is to be expected as the reducing components and enzymes and cofactors that are responsible for the reduction mechanism are being consumed and passive antibody reoxidation is occurring. So within the literature, there's been a lot of articles and investigations for the intercellular enzymes in CHO cells that are involved in the reduction mechanism of antibodies in cell culture. So with the two main enzyme pathways here, thioredoxin and glutaredoxin pathways, we see the enzymatic flow chart here representing all the different enzymes and the cofactors involved in the antibody reduction mechanism. So it begins on the left with the ATP and glucose-6-phosphate dehydrogenase being responsible for the replenishing of the NADPH from NADP. Those electron transfers are then responsible for reducing the thioredoxin reductase and thioredoxin, which ultimately lead to antibody reduction. In the bottom, a very similar electron transfer is occurring with the glutathione reductase pathway, here with glutaredoxin being another alternative enzyme that's responsible for controlling redox potential with the cell that can ultimately lead in antibody reduction. So these two pathways here, the thioredoxin and glutaredoxin, are released from the cells, and what our hypothesis is that depth filtration is causing cell lysis, releasing these enzymes into the clarified harvest, leading to monoclonal antibody reduction. We do also have internal evidence, which will not be discussed today, that thioredoxin reductase can also reduce an antibody directly in an NADPH-dependent manner. So the investigation of depth filtration parameters in antibody reduction, the schematic of our investigation is shown here on the top, where clarified harvest was pumped through a depth filter, a final 0.2 micron filter for clarification, and as an investigation, we were able to take filtrate samples, so however many filtrate samples we had taken throughout the course of depth filtration, and then also a bulk clarified harvest here represent the entire pool processed through the depth filter. These samples were then stored in tubes that were sealed with and without headspace. Here we've got no headspace, and stored for a predetermined amount of time, up to 1 week at 2 to 8 degrees for characterization and analysis. Going back here, the filter types that we had investigated here were the two filters that we have in our platform, so 10SP O2A and a 30SP O2A. These filters are manufactured by 3M, and 25 square centimeters was our effective filter area that we had used. The flow rates were 25 to 100 liters per meter squared per hour, or LMH, and throughputs of up to 150 liters per meter squared. So in these studies, two antibodies, MAB-A and MAB-B, these are IgG1 and an IgG4 antibodies were investigated in these studies. The monitoring parameters that we had used to understand the characteristics of the filtration are listed here on the slide. Lactate dehydrogenase is an indicator of cell lysis. We use the CDEX Bio HT as our measurement here. Free thiol content, this is a sulfhydryl-containing measurement that we had used to estimate the total amount of sulfhydryl-related reducing components in the clarified harvest. And intact MAB purity was measured by non-reducing capillary electrophoresis using the caliper GX2. what we have here is on this initial slide here. So the next few slides are just going to discuss some of the highlights from particular experiments and parameters that we had done during the depth filtration unit operation investigation. So here on the left-hand slide is an example of a differential pressure trace for two extreme filtration conditions for monoclonal antibody A and B. So we see the differences in differential pressure traces between the two, and then reaching maximum pressure, delta P, of approximately 30 psi at different stages of the depth filtration throughput. So with MAB A, we reach approximately 100 liters per meter squared, and with MAB B, we get to approximately 180 liters per meter squared. The characteristics of the cell culture as a function of throughput are shown on the right-hand side. The LDH measurement is on the top, and the thiol measurement is on the bottom. So we see that both of these attributes, LDH and thiol, both increase as a function of filtration throughput. They also show that the higher differential pressure, which was also observed with MAB A, is elevated above what we observed with MAB B. So we do see corresponding attributes for both of these LDH and thiol measurements, indicating that we see indicating cell lysis throughout the course of depth filtration, as we see with the increase in LDH, and then also the increase in thiol, indicating release of the reducing components into the clarified harvest. Similarly, these samples were then staged and analyzed by capillary electrophoresis. So the light gray bars represent the time 0 analysis, or day 0, and then the day 7 analysis are represented in the darker gray bars. So we see with the MAB A, which is shown here on the top, that antibody reduction is observed at approximately 88 liters per meter squared, and also approximately 120 liters per meter squared. The MAB A, again, is the one that reached higher differential pressures earlier, and MAB B, when shown on the bottom, we don't see antibody reduction until we get to approximately 162 liters per meter squared, as shown on the bottom here. So the data indicate that there's a throughput for volumetric throughput threshold for each system based on each of the filter pools after extended storage. Interestingly, the thiol and LDH correlation plots are shown here. So within each correlation of thiol and LDH, MAB A and MAB B both have positive correlations between these two attributes, indicating the release of LDH and then also the release of thiol, or correlative measurements to determine cell lysis and release of these components into the clarified harvest. The higher LDH and thiol content also correlate with MAB reduction. So with these data shown here, the LDH and thiol content of the clarified harvest can be used as indicators for quality. The graph shown here on the left is LDH, and then on the right is thiol. Plotted against these two attributes is the intact monoclonal antibody as measured by non-reducing CESDS. We do see, as a function of each of these contents for LDH and thiol, there is another threshold that we're observing that reduction is happening within a certain period, a certain measurement of both of these attributes. So for LDH, we're seeing antibody reduction occurring when the LDH levels reach approximately 8,000 to 9,000 units per liter. Similarly, with the thiol content, we do see a similar threshold where the thiol content indicates that 600 to 800 micromolar thiol is where we start to observe antibody reduction. The filtration flow rates, as previously discussed, was also a parameter that we had tested in our depth filtration unit operation investigation. So here we have filtration fluxes of 25, 50, and 75 liters LMH on a 10 SP filter that was tested. Again, showing the attributes of LDH and thiol on the left and right-hand side respectively. We do see an increase in both of these attributes as a function of throughput, but also within each system, we do see an increase in each of these attributes as a function of filtration flux. So the lower stars, asterisks represented on each of the graphs, show that the lower filtration fluxes release and have less cellular lysis, as shown here with both LDH and thiol measurements. As we increase the flux, we do see a corresponding increase in both of these attributes of LDH and thiol. So these attributes increase with filtration flux and likely due to the increased cell lysis at the higher filtration fluxes as a result of higher differential pressures. These filtrate pools were then stored for the similar period of time with no headspace. So we do see with the graphs here for 4 different filtration fluxes of 25 to 100 LMH, we do see that the 10SP filter, which is a larger pore size filter, that is generally treated the cell lysis and more gentle as compared to the 30SP filter. So as we store these materials for up to 7 days, we do see antibody reduction occurring quicker in the 30SP conditions. The 10SP conditions up to 50 LMH and in part 75 LMH maintain purity in each of these filtration product pools. Also, we do see an increase in antibody reduction as the function of filtration flux, and this was observed with both filters. The 30SP resulted in lower purity quicker, and that is likely due to the increased pressure with the smaller pore size filter characteristics of the 30SP depth filter. So one thing that we are trying to bring this all together with is differential pressures. So higher differential pressures during the depth filtration unit operation can lead to increased cell lysis. So the delta P is considered to be an aggregate of filter type, flow rate, and throughput. So we do see on the left-hand side just a snapshot of some of the depth filtration differential pressure traces that we had during this investigation here. On the right-hand side, we see a depth filtration delta P's plotted against LDH and thiol. Both of these attributes represent a linear relationship up to about 15 psi as a function of delta P. Beyond 15 psi is when both attributes of LDH and thiol begin to plateau, and this is not surprising since we are in the exponential increase phase of the depth of the differential pressures for each of these filtration conditions that we had tested. Finally, the differential pressures leading to increased cell lysis, leading to MAB reduction. What we have here is an aggregate of data from both MAB-A and MAB-B for all the intact MAB purities that we had tested throughout this investigation, plotted against delta P. So we see, aside from the one data point at approximately 7 psi, we see all the samples that were tested and taken below 10 psi throughout the course of the filtration, measured at high levels of purity. Once we begin to cross this threshold of approximately 10 psi, we do see a very stark decrease in MAB purity after the extended storage. So this is indicating that the reduction in samples is observed above 10 psi. Now again, these samples represent the PSI, or the differential pressures that the sample was taken at throughout the course of the filtration. So this is indicating that the delta P is releasing and causing cell lysis, releasing these components, and ultimately leading to monoclonal antibody reduction over extended periods of time when the differential pressure traces get above 10 psi. In conclusion, what we had done was observed antibody reduction in large-scale processing of the 2,000-liter bioreactor. A risk mitigation strategy that we employed using an active air overlay in the clarified harvest tank was successfully implemented and prevented antibody reduction in our GMP production batch. Careful consideration should be given when using single-use technologies in the consideration of what type of oxygen maintaining system is used and how it applies to single-use technology, specifically bioprocess storage bags, and also the use of disposable depth filters as a means to clarify cell culture harvest. The high differential pressures that we have observed during depth filtration was identified as the root cause of cell lysis, leading to antibody reduction. The filter flux, throughput, and type are critical in controlling cell lysis, and we have demonstrated strong correlations of delta P with our product quality. Product quality meaning LDH, thiol, and ultimately the intact MAB percentages measured by non-reducing CE-SDS. So with that, the filtrate quality should be monitored closely during the development of the recovery unit operation of CHO cells during disposable depth filtration. So with that, I'd like to acknowledge and give thanks to all the folks here in Seattle at our process development facility from both downstream, upstream, analytical, and also our BPD management here that allowed us to further investigate this and provide some risk mitigation levels to maintain clarified harvest integrity and to maintain intact MAB.
Thank you, Brian. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the Questions tab located directly below your webinar screen, and we'll go through these at the end of the webinar. Now, please allow me to introduce our second speaker, Alexei Voloshin. He is the Global Application Strategy Specialist at 3M Separation and Purification Sciences Division. He is responsible for product application strategy and manages technology projects, collaborations, and alliances around the globe. Alexei brings more than 15 years of experience in bioprocess development, as well as technical, operational, and business knowledge in the biotechnology space. Before joining 3M, Alexei led molecular diagnostic programs at Theranos, Inc. He is a founding member of TheraOne, Inc., a startup company focused on frontline cancer immunotherapy. Before TheraOne, Alexei was an early member of Sutro Biopharma, Inc., a company focused on development of novel biologics and bioprocesses. There, he was responsible for technology strategy and process development. He has authored a number of publications and presentations dealing with novel bioprocesses and their applications to making advanced biotherapeutics. Alexei got his PhD in chemical engineering from Stanford University and Bachelor of Chemical Engineering and BS in Computer Science from University of Minnesota, Twin Cities. I'll now be handing over to our second presenter. Welcome, Alexei.
Thank you, Stephen, and thank you, Brian, for a great talk. What I'm going to try to do is complement what you presented, Brian, with some perspective from the sort of technology point of view of depth filtration, and perhaps shed some light or perhaps evoke even more questions on on how the observations that you've made in purification of your monoclonal products are related to the actual technology of depth filters and their utilizations in the process. To the audience, good morning, good afternoon, and good evening, wherever you are, and thank you for your time. So if you look at the basic structure of qualification trains, there are a number of different strategies that one can use. However, all of these strategies have about the same structure, and the structure is essentially composed of the primary cell mass separator stage, and there are a number of technology and approaches that people have successfully utilized. But it's interesting that regardless of what primary cell separator stage you use, there's always the second stage, and very frequently that also requires at least some sort of a depth filtration approach plus a membrane approach to then protect the sterilizing membrane. And the reason why that is so is that each one of these primary separation stage approaches, they always leave these relatively small by depth filtration, by large by membrane filtration particle tail. And that particle tail, as it turns out, and as you will see in this presentation, is composed not only of perhaps particles that are already present in cell culture, but also contain artifacts of how people design and implement the primary cell mass separator stage. So you're essentially cleaning up the problems that you create in the first stage by the second stage. So for the purposes of this presentation, we're going to focus on the primary depth filtration stage. So this is now the cell separator stage, which is now implemented as as depth filtration. And if you look at the primary goals of that stage, there are only really two goals. Number one is to capture the cells. Number two is to capture very, very large cell debris contaminants, perhaps shrunken cells or dead cells, depending on how the cell culture strategy is being executed in terms of viability of the cell culture itself and in terms of particular biochemical characteristics of the cell culture. So if you look at, for example, 3M's product line, and if you then disassemble and you look at the primary filter grades that are utilized and that are designed to be used as a primary cell capture stage, there are 4 of them. They span everything from 0.1 SP, very, very open grade, all the way to 30 SP. And so these grades span the approximate or nominal pore size range, where the cells are most likely going to be captured by the pores themselves. There are a couple of things that are very, very important to understand. Is that number one, depth filtration is not membrane filtration, which means that the basics, physics of their manufacture is the fact that you have a very ill-defined pore size, and there's a large distribution of pore size. When somebody, for example, tells you that, or gives you a 0.2 micron filter, that 0.2 micron membrane filter has a very tight pore structure simply because that's how it's manufactured, and that's how it's produced. When somebody gives you a harvest depth filter, that particular media, which is made by wet laid process that's very, very similar to how paper is made, has a very, very ill-defined pore structure that that spans a very, very large pore size range. And that is both its blessing and a curse. The blessing is the fact that it's able to deal with highly heterogeneous as well as also ill-defined streams and to be able to clarify them. The curse is as Brian pointed out, that you get to run into some of these artifacts that have to do with the fact that there are a number of different pore sizes. The pore sizes have different tortuosities to them, and so such one has to be able to take that into account. You can also see here on this graph that, for example, most of these grades, the pore size is definitely less than the hydrodynamic size or the light scattered size of a cell. So a typical cell for CHO will be somewhere in the range of, let's say, 14, 16, even 20 microns in size. And obviously here, the bulk of the pores themselves, as illustrated in this graph, are smaller. And that's not surprising. In order to capture the cell, you have to be able to capture it sort of in a slightly smaller pore, such that it actually gets trapped in there rather than pass through. So what we're going to do is that we're going to look at the performance in terms of clarification of these filter grades individually, to be able to understand where's the threshold between being very gentle on these cells and being very, very rough in causing lysis. So what we did was that we conducted a couple of very, very simple experiments to be able to demonstrate the major characteristics and perhaps the major problems that are associated with operation of depth filters. So in this slide, I'm showing very, very simple experimental setup. So what we do is that we grow a culture that's essentially 99% viable. And the reason why we want to do that is because we want to have a very low background that we're measuring against. We want all the cells to be intact. We're not very much concerned about the density, and in fact, slightly lower density of cells in the cell culture. And you can see here it's approximately 6 million cells per centimeter cubed. Actually helps us because all of these effects then occur in slow motion, and you're able to observe them in great detail level. If your culture is very dense, then a lot of this happens very, very quickly, so it's very difficult to observe. But in our case, it's easier to observe because these effects happen very, very slowly as you keep pumping liquid in. Then what we do is that we take each one of those filter grades and we put it as a primary stage, and we clarify the cell culture at 120 LMH. and we go up to either 15 psid, at which point we say, look, this is really the largest threshold in terms of pressure that we want to run into, because at that point you're really at the diminishing return stage, or that we break through. In which case we say, look, if you get up to 100 NTU in terms of turbidity, it'll be very interesting to look what's in that turbidity. Do we have intact cells? Do you have fragments? Do you have other things? But for all practical purposes, we're done simply because the filter is no longer effective in terms of clarification. And then we utilize three different assay methods to try to understand what's coming through the depth filter, so what's coming on the other side. And this is turbidity, dynamic light scattering, as well as Coulter counter. So dynamic light scattering, Coulter counter method, they are sort of complementary. One allows us to watch for fragments less than 4 microns in size. The other one allows us to watch intact cells and fragments that are greater than 4 microns in size. Then what we did was that we really created 3 pools. 0 to 5 psid, so very, very low pressure that we expect not to have a lot of cell damage. Then 5 to 10 psid as the pressure rises in the filter, and then really very, very high pressure, 10 to 15 psid, and really look at what's happening in each one of these pools to try to understand how does the makeup of the filtrate actually change. So if you look at the basics of throughput and turbidity across each one of these depth filter grades, you can see some very interesting things there. For sure, not all created equal. And you can see that, for example, 0.1 SP and 0.5 SP, the turbidity is very low throughout their entire throughputs. And this has to do with the fact that these are very, very gentle grades, such that, especially in a culture which is almost 100% viable, there's really not a lot of debris because everything is intact. And this is exactly what you observe here, is that really you're able to very, very gently pack the cells at very low pressure into the actual grades and have a very, very clear liquid left behind them with very little debris. What happens once you run out of this bed volume in these filters? It's very, very simple, and you will see in the next slides. The cells simply break through. So you're essentially operating a size exclusion bed in a size exclusion chromatography mode. As the bed fills up with the cells and you push more cells in, the cells simply start coming out on the other side. But what that gives you is that gives you ability to run a very, very low pressure system that's extremely gentle on the cells themselves. As you then move to 10 SP grade and then 30 SP grade, you see that the situation changes. Is that, for example, 10 SP, as the pressure starts to go up on it, you start seeing turbidity coming through. Clearly, that turbidity, based on our experience from 0.1 SP and 0.5 SP, has nothing to do with the cell culture itself. The turbidity is essentially cell mass being sheared inside the filter. And so this is the artifact that I was talking about in the beginning of the slides, that your first stage is starting to create issues for your second and third, however many stages you have before your material ends up in the intermediate storage container before going onto the capture column. And you can see that as you reach 10 to 15 psi, you can see that the turbidity is very, very high. In fact, we had to stop it slightly earlier simply because we already reached our 100 NTU limit, and there was no reason to keep extruding the cell culture through that filter. In the case of 30 SP, it's tighter, and so that transition area in terms of the pore size that's capable of cell extrusion is smaller. And so you still see, for example, if you look at the turbidity on the 30 SP, you still see the turbidity going up, but as the filter pressures out, its turbidity stays relatively small. And this is, I will show you a little bit later why that is so, but in summary, it is because simply the pores that are large enough to be able to extrude cells fill up very quickly, and so the filter essentially clogs. It becomes almost like a membrane filter as far as cells are concerned. So you can see here that there's a transition right around 0.5 SP to 10 SP area from being a grade that's very, very gentle and is very, very well sized for a typical CHO cell to a grade now which has a lot of pores inside it which are sized to shear the cells themselves. So you can look at the culture counter method. So this is where we're watching essentially cells in large fragments. You can see the feed of 99% viability, and so our cell size is somewhere around 15, 16 microns, so pretty typical for a CHO cell. And you can see in the shear field, somewhere around 5 to 10, 12 microns, there's really not a lot of things there. There are really not a lot of fragments. And so this is, a very nice thing to have because your background noise is very, very low. So then as you go through 1sp or 5sp, and so these are the first fractions of 0 to 5 psi, and 10sp, you can see that they're all very, very similar. You can see that the shear field is slightly increasing in the 10sp. You can see there's virtually no increase. If you look at the fragmentation pattern there towards the left side of the graphs, you can see the fragmentation pattern is, for all practical purposes, identical to the feed, so you're not shearing at all. Whereas the 10sp is starting to shear. And so far, if you look up to 5 psi, all of these filters do a pretty good job of keeping cells intact. So then, as you move into the 5 to 15 psi fraction, you can now start to see the 10sp really starting to shear the cells. So I'm not showing here the 01sp and 05sp because the 01 and 05sp never get to that pressure differential, they break through. I'm not showing the 30sp because 30sp is tight enough such that these pressures, it doesn't allow any of the debris of that size to come through, so your graph is essentially empty. But the 10sp is very interesting because you can see that it's sized in a way, that grade, that once your pressure really clicks above 5 psi or so, it really starts to shear. So you're really forcing, the pressure is forcing the cells into these pores that are capable of extruding the cells themselves. And all of a sudden, your shear field on the left side of the slide blows up compared to the feed. And so this is what we believe is one of the, is the major reason for example, observations that Brian presented, is that now you are really creating a lot of extrusion in the media itself. So then you can sort of put the data together just for 10sp, and you can see kind of an interesting progression, right? 0 to 5 PSID, very good. As soon as your pressure clicks over 5, you start getting a lot of shear, and that shear really continues all the way up to 15 PSID. So in this case, if you are utilizing that grade as part of your regimen, it is obviously important to keep the shear, the pressure differential across that grade at less than PSID. But you can see that in general, it is a grade that's relatively dangerous to use. So then you can look at, for example, dynamic light scattering of 10sp, and you can see what's coming through at very, very small sizes. So this is really below a micron or so. And so this is a technique where you shine a laser inside a clarified fluid, and you're looking at Brownian motion of particles that you can then back out their sizes. And so one thing to understand here is that large particles obscure the signal from small particles because they create a lot of shadowing. And so you can really tell how clear your fluid is by how many small things you can see. So for example, at 0 to 5 PSID, you can clearly see your map at about 12 nanometers in size. And so that tells you that there just isn't a lot of large things, things that are larger than map. You can see that there clearly are, but there's not a lot of them. But as you go to 5 to 10 PSID, as you go to 15 PSID, you can see very, very quickly that your map gets obscured very, very quickly. In fact, a lot of things that are bigger than map, like for example, genomic DNA peak at 100 nanometers, also gets obscured by the time you get to the 10 to the 15 PSID fraction, and that gives you an idea that you're really dumping in a lot of very, very sort of large contaminants, probably from the cell shear into that range of submicron size or near micron size. You can see here for reference O5SP, so this was taken, this spectra was taken at the O5SP, so this is a real-time spectra taken at the end of the clarification, so right before the cells were breaking through the bed. And you can see there that the feed is pretty clean. The feed is, you can very, very well make out the MAD peak there. It's completely resolved. So you can see that the situation is somewhat similar to what you get on the 10SP at 0.5 PSID. And so that gives you an idea that really, both of these grades, as long as you're running at very low pressure, you're utilizing the pores in the size that are very, very gentle on the cells, which means you're not extruding the cells to the filter itself. But obviously, as you build up pressure, the 10SP starts to then fill up its sort of cell extrusion zone in terms of its pore size. So then you can look at dynamic light scattering of 30 SP. So 30 SP did not produce any signal above 4 microns simply because it's too tight. But we can watch under a micron or near a micron range to try to understand, are we getting an extrusion in that size range? And as you can see clearly here, yes, we do. Is that if you look at below the micron size range, you can see that, for example, 0 to 10 PSID fraction here, you can sort of start to resolve your MAB and everything is looking good. But at 10 to 15 PSID, you can't really see your MAB almost at all. Maybe that peak is there and that mess that you can see on the dynamic light scattering spectrum, but it's very, very difficult to tell. So just because you don't see a lot of these very large fragments that you can sort of very, very easily tell on the culture counter, the fact that you're breaking the cells and they're not showing up on on a lot of turbidity, and you're not picking them up as a lot of turbidity on your turbidity screen, doesn't mean that you're not extruding, and doesn't mean that you are not actually dumping in all of the enzymes as well as obviously these reductases into your cell culture fluid. And again, I have the O5SP dynamic light scattering spectra here as a reference, just to show you the difference between running a filter that's very, very well sized to be a gentle filter for the cell capture versus something that's much, much more tight. So if you look at the LDH assay, which is one of the things that we've also utilized in this, and you look at the fractions now for each one of these grades, you can see that something very interesting. Number one is that O1SP and O5SP, they do not release any LDH that was not there in the feed already. So the feed signal is at the very, very right of the slide. And so you can see that these grades are so gentle that there is really not much share going on, at least you can very easily pick up using this method. You can see that the LDH really blows up in these middle fractions, both for 10SP and 30SP, and that's not surprising, as we saw in the dynamic light scattering and culture counter analysis. That's to be expected. We're extruding a lot of cells. What's interesting is that your level of detectable LDH falls off at higher pressures. And so that's we will revisit this in a couple of minutes after I show you some other data that perhaps this will become evident why that may be so. But it's interesting that this danger zone of really 5 to 10 psi is really, really evident here in terms of the signal of the LDH activity. All right. So now we can what we can do is that we can after analysis of these grades, we can now go to and look at the actual dual layer products that we make for biopharma industry, which form these deep funnels, and how is it now that armed with this knowledge, we can now understand how each one of those grades behave. So Brian went over the performance of 10SpO2 and 30SpO2. So 10SpO2 has the front grade being O5SP, followed by 10SP. 30SpO2 has the front has O5SP followed by 30SP. We're also going to study the 5SpO1, which is also a product that we make that's composed of these very, very gentle of the most general grades, and then also one slightly tighter product that we have, which is 60 SpO2, and that's something where we combine a very, very open 05 SP with a very, very tight 60 SP to try to be able to give you really single-stage, all-in-one solution for clarification of both large cells and large fragments, as well as the filtration of small fragments, that may be present, for example, if your viability is not very high. So, similar type of approach. What we did here, it was that we tried to create a very simple setup here, where we also took high viability cell cultures. So this is slightly different cultures, slightly different, higher cell density, 98% viability. We're also running a depth filter, one of these depth filters at 120 LMH. Where we are screening for being either below 15 psi or 100 NTU behind it. We also put a 0.2 PES behind it as sort of a turbidity trap, such that we can observe using a simple pressure device whether the depth filter is breaking through. And we have ability to sample both right after the depth filter, such that we can see what's coming through it and hitting the membrane, or we can actually also sample after the membrane to look for soluble contaminants such as LDH, as well as the product quality itself. And so you can see here, again, throughput and turbidity. You can see the data here is in some ways very interesting. You can see that 5 SpO1 and 10 SpO2 really produced about the same throughput. You can see that 30 SpO2 and 60 SpO2 are slightly lower, but in none of these cases are you producing 4 or 5 times the throughput. All of them are about the same, which tells you that choosing a more open grade or a tighter grade isn't always straightforward when you're using cell capture. It's really how you approach both your cell culture, but also how well is the filter grade itself tuned to your cell culture characteristics. So it's not always straightforward. As you can see, turbidity, again, 05-SpO1 produced very, very low turbidity up to the breakthrough of the intact cells through the filter. You can see the 10-SpO2 produced very low turbidity in its low pressure fraction, and very high turbidity in a high fraction. And that's not surprising. There's a 10-SP grade in there. And what we've seen is that 10-SP grade produced a lot of shear, a lot of turbidity. Also, 30-SpO2 produced a better turbidity control, but nonetheless produced much more turbidity in its later stage, probably also through extrusion. And one thing that's very interesting, you can see that 60 SpO2, low pressure, high pressure, produced about the same turbidity, and we will see the reason why that is. So if you look at clarification dynamics using culture counter of 05-SpO1, when your bed has not filled up yet, when you still have empty pores inside the bed, you can see there's nothing coming through the filter itself in the size range of really 4 to 34 microns, and that's not surprising, simply because you're able to capture all the cells very gently, and because there's no shear, you're not generating any particle shear field, so you get a very, very clean separation. You can see that as the terminal event is reached, and there's a breakthrough of the turbidity, and if you look inside that turbidity using culture counter, you can see that it's all intact cells. The shear field stays effectively identical to the shear field in your feed, which means that you're not shearing any cells at all. If you're looking at 10 SpO2, you're now getting at 5 psi, and here we reached our endpoint 11 psi. You're now getting a lot of shear field. Also at low psi, you're not getting any cells breaking through at all. So that's your 05 SP. Front grade working really well. As you build up the pressure on the filter, the 10 SP starts to shear the cells that are breaking through 05 SP into a 10 SP grade, and the 10 SP starts to shear under pressure as it quickly fills up. And so that's the reason why this grade is somewhat tricky and dangerous to use, because it's very, very easy to generate shear in that grade. 30 SP, you can see here that in a culture counter method does not produce anything, simply because 30 is too tight. It will shear the cells, but it won't. But that shear, the extrusion through the pores will produce a much finer particle size such that you can't really see it on using this method. 60SpO2, same thing. Whatever it does, it's just very, very tight, so it doesn't produce any debris fields simply because the pores in the 60 end up catching everything, and so you won't be able to see any particles that are in that size range. You can see that, for example, at terminal events, when your filter's at the end of life, the spectra on the sub-micron range, now endemic light scattering, are very, very different. 5SpO1, you can clearly distinguish the peak. 60SpO2, you can clearly distinguish the peak, and these are very sort of qualitatively different results that you get from 30SpO2 versus 10SpO2. And this is, again, because 60SpO2 does not produce any shear because 60Sp is just too tight for cells to come in. So a cell sees it essentially like a membrane, and 5SpO1 simply breaks through as the cells fill up. 30SpO2 and 10SpO2 produce shear in that intermediate pressure range. So if you look at the dehydrogenase, the LDH assay, you can see again that the two filters that shear the cells, produce very, very similar data to what Brian showed. That yes, you do get a lot of LDH dumping into the culture, and obviously this is also an indication that there are other enzymes such as reductase, glutathione reductase that are probably also making it in the clarified cell culture fluid together with your antibody. This is not the case in 5SpO1 or the 60SpO2, simply because one is gentle and the cells break through. The other one simply pressures out at the end just like a membrane, simply because 60Sp is too tight for the cells to come in at all. And so they both produce essentially background amount of LDH is very similar to what's already in the feed coming into the depth filtration in the first place. So if you look at, so what we did was that we conducted a very simple analysis in terms of product quality. The MAB line, which we're using here, produces a very, very stable biosimilar, so we're unlikely to see a lot of reduction here from the get-go, but we looked nonetheless. And what we did in order to make things simpler was that we treated the clarified cell culture fluid with iodoacetamide. Iodoacetamide is a very good and simple to use alkylating agent, which caps all of the reduced sulfhydryl groups, both on the enzymes themselves, such that we're able to deactivate the enzyme, the reductase activity, as well as capping the sulfhydryl groups on the reduced antibody fragments themselves, such that now the system is frozen in time, and we can run a denatured gel here and see what happens. So here is pretty much our about 150 kD antibody. It's an IgG1. And if you zoom in at 50 kDa and 25 kDa region, you can see that in the lanes of 30 SpO2 and 10 SpO2, there's a lot more bands. And we believe that at least some of these bands here are essentially alkylated, capped light chains and heavy chains. We have not confirmed that using a Western blot or any other way, but you can see that the lanes produced from the 10 SpO2 and 30 SpO2 are qualitatively different in terms of the different bands that they contain from the feed, which is lane number 1, from the 5SpO1 filtrate, which is lane number 2, and 60 SpO2, which is lane number 5. And this also kind of mirrors what Brian presented, is that yes, there is a significant dump of both HCPs, and probably also product-related contaminants, which are reduced now into your cell culture fluid. So if you go back, I have 1 minute here to discuss this kind of interesting problem. Why is LDH lower in fraction 3 compared to fraction 2? We can revisit that activity, revisit that question in 10SP and 30SP. What I hope that we can sort of at least hypothesize here why that is true is because if you consider a porous bed of distributed in size, and let's say your green pores here are the pores that are gentle for cells capture, the yellow pores are the ones that shear and extrude the cells, and the red pores are pores too small for the cells to enter, you will see that as you put more material through, first you have the large pores fill up. And so there you don't get a lot of shear, don't get a lot of LDH dump. Then the pores that are slightly smaller, that are now extruding your LDH out of the cells, are now filling up, and that's why you get the signal. But as those fill up, the only pores that are left are too small for the cells to come in, so you're starting to just accumulate the cake. The filter essentially behaves as a membrane filter as far as your cells are concerned. And so your LDH activity starts to diminish because you're simply not breaking any more cells inside the filter. And so that's the hypothesis why we believe that at higher pressures, the amount of LDH actually starts to go down as far as the signal itself. So as far as conclusions, is that what I hope that I was able to elucidate and also support Brian's presentation is that cell shear is an important parameter to consider during clarification train design, simply because these systems are complicated, and the depth filtration media is a media that's made on a process that produces approximate results. It's not a highly defined technology in terms of its pore size. And obviously, product quality is very important. It affects your entire downstream process. It affects the productivity, and so certain strategies always need to be made in order to be able to mitigate this shear and to be able to really tune your clarification system to the cell culture itself. So, for example, as I've shown you, Zeta Plus 5-SPO1 is very, very gentle, and it operates more like a size exclusion bed. That's really sized by cell breakthrough more than anything else. And a lot of these tighter grades can still be used, but you need to be very, very careful in operating them at low pressure to avoid that extrusion and really accessing in that pore size that extrudes the cells. So with that, I would like to thank you for your attention, and we'll take any questions.
Thank you, Alexei. We will begin the question and answer segment of the webinar, but once again, I would like to remind our audience that you can still send in your questions via the questions tab located directly below your webinar screen. So now, our first question is, how does the quality of cell culture relate to these observations? For example, the percentage of variability or cell density?
This is Brian speaking. That is something that we have not investigated and is kind of on the list for subsequent studies. In terms of viable cell density and the way these cells behave in cell culture, I think one of the interesting things to look at would be the integral cell density, or integral VCD, as a function of bioreactor, because these two mechanisms there's two different mechanisms of cell death. There's apoptotic and necrotic cell death. So, measuring the VCD would be interesting, but as it relates to integral VCD, and then also the total cell densities, total cells coming through the filter, some other correlations could probably be elucidated in that analysis as we start to control bioreactor conditions a little bit better and get a better understanding of that.
Thank you.
Yeah, I'd like to chime in on that. I think Brian, I think what you're saying is sort of exactly right. Is that if you look at the basic physics problem, is that you're trying to capture cells intact inside a porous bed. Thus, your VCD will be the controlling parameter, simply because it's conservation of volume, and so the larger your VCD, the more simply bed volume you're going to need in your depth filter, because there are only two ways to do it. You're either keeping cells intact, in which you have to account for the entire volume of the cell, or you're breaking the cells, in which case you are essentially draining the water and whatever is inside the cell. So there's always going to be trade-off between whether you would like to be able to keep more cells intact, or you would like to be able to compress that cake even more simply by rupturing the cells and draining the water out of them.
Yeah.
Thank you both for your answers. Our next question is, was a larger pore size evaluated with the 05A?
In our particular case at BMS, no, it was not. We had 10 SP and 30 SP filters available in our inventory. And again, this would be another subsequent follow-up study, and I think Alexei's data, which was a very good presentation on the characteristics and the behavior within depth filtration, the wider pore size filter of the 05 would be a really good way to go to controlling this a little bit better during the depth filtration unit operation.
Our next question. Are the shearing effects specific to 3M products, or is this a characteristic that is inherent to all of the depth filtration technology?
I think that this is probably inherent to the technology itself, simply because regardless of whether it's us or our peers, everybody manufactures the depth filtration media using very similar wet late process. I think the biggest difference that you're likely to find among manufacturers of depth filters is the pore size range that is offered in the product lines. So, for example, at 3M, we have a very, very wide range of these filters and pore sizes, and so it's not sort of a matter of whether somebody makes gentler pores than other things because of manufacturing. It's simply does your particular supplier have the pore size and the pore distribution that's the best matched or well matched to the characteristics of your cell culture and the size of the cells themselves.
Thank you for your answer. Our next question. How does the flow rate affect the shearing inside the depth filters?
My understanding is that the flow rate primarily determines the pressure. The cells themselves are large enough such that you're still going to be diffusion limited in terms of pore diffusion, regardless of what flow rate you go. Simply because cells are very large compared to the pores themselves. Is that you're not going to have pores inside your depth filters which are 20, 30 times the cell size. Such that that's not the problem. I think that the biggest driver that's determined by the flow rate is the pressure differential. So you're much more likely to reach this extrusion pressure quicker if you flow faster. So that's probably the most the biggest variable that the flow rate affects. What do you think, Brian?
I would agree with that, Alexei. Yes, thank you.
Our next question is for Brian. For your second GMP without reduction, was air overlay the only mitigation factor?
No, it was not. We had, during the second GMP production batch, the air overlay was the change that we had implemented for clarified harvest storage. The second mitigation or factor that or strategy that we had implemented was we did reduce the flow rate during depth filtration. So we decreased it by approximately 1/2. So that was based on the pressure, like Alexei said, as reaching the maximum pressure quicker. We decided as a supplement to the air overlay to decrease the flow rate during depth filtration by approximately 50%.
We have another question for Brian, but Alexei, you feel free to chime in. Were you actively controlling DO during clarified harvest hold? What is your strategy for designing scale-down model of recovery steps, delta P and DO as controlled parameters?
So if I understand this question correctly, so for the DO being controlled in the clarified harvest, this was something that we controlled DO and we actively monitor it during the entire hold during the second GMP production batch. That was not necessarily something we needed to turn up or down. It was a control that we had maintained and monitored during the entire storage. Regards to the follow-up question for the strategy of designing the small scale-down model of the recovery step, the delta P was just an output that we got what we got. This was something that we had varied these tunable parameters in the depth filtration, like the filter throughput, the flux, and the pore size, or the filter type that we use, and our delta P was just an output. Using DO, for the data that was shown here in this presentation, the air overlay in our small-scale storage vessels could not actively be done. So we just had to put in a headspace of sufficient volume to maintain that oxidative environment. Other studies that we had done in the lab that were not in this presentation, we were able to store some of our larger volume clarified harvest in vessels that were used for upstream production of bioreactor vessels, where we were able to control, monitor, and trend the dissolved oxygen. So the data that was obtained from those larger volume holding vessels did corroborate well with the data that was shown here, showing that the active air overlay or the headspace conditions resulted in the same intact MAB purity.
Thank you for that. Our next question. How do you know that the release of Thiol and LDH cell lysis is caused by the flow rate through the filter and not due to the higher pump rate?
This one is for me again. I would say I can't say 100% sure, except in these studies that we had done both at lab scale and during production, we had used both peristaltic pumps and a lower shear quattroflow pump. The peristaltic pump was done in the lab scale, and then we also performed the quattroflow pump at the lab scale. The quattroflow pump was used during GMP production. So similarly, we did see similar trends in delta P using both of these pumps during the clarification of cell culture harvest. So Alexei, you might want to chime in on this, but the delta P and the cell lysis is primarily being contributed by the depth filter. The type of pump and the little bit more aggressive nature of the peristaltic pump may be a factor in cell lysis, but in our studies we showed that both pumps showed a similar cell lysis and trends with LDH and antibody reduction.
Yeah, I agree. I think that in principle you can always find a pump where you could get a lot of shear, but in this case you're primarily using pumps which are designed to be very, very low shear, and so that contribution is probably going to be very small, if anything, compared to the extrusion through the depth filters at high pressure.
Right.
Thank you. Our next question, I believe this one is for Alexei, is did you manage to reproduce this reduction on small scale? This was done on small scale, so reduction is so that experiment was done at relatively low scale of hundreds of milliliters. So yes, it is reproduceable.
Yeah, and I might add to that too. I think a lot of this has been kind of a hot topic, and a lot of people have observed antibody reduction, across the industry, and sometimes it doesn't rear its ugly head until large-scale production batches. During, obviously, with the data that we showed, it came up during our large-scale production batch at a 2,000-liter single-use bioreactor. But with a lot of the data and supplement studies that we had done, we had quality cell culture material from various scales, from 50-liter, 200-liter. Down to 2- and 5-liter bioreactors, and even 3-liter shake flasks as well. So we were able to generate similar data, regardless of scale, when we started normalizing and investigating this depth filtration unit up.
Yeah, I think one thing I think that's important to realize, I think that when people started seeing it at a large scale and they couldn't see it very well at small scale, at least in my observations, had to do with the fact that it's very easy to oversize a filter at small scale, simply because, instead of one capsule you put two, and then there's no problem because you're never actually running it up to high pressure. Whereas at, large scale, you're trying to minimize your footprint, so you're trying to get that last performance out of the depth filter. And so the depth filter is utilized a lot more aggressively at large scale, compared to your typical prep work that you would do at small scale, where your filter is always comfortably oversized. And I think that was a little bit of a disconnect when people started observing that. But then as people started building much more accurate models and actually pushing the depth filters at small scale to as hard as they did at large scale, then they were able to reproduce the effect.
Mm-hmm.
Thank you.
I agree. And one other thing to add to that too, sample handling is very key to observing this antibody reduction, and like Alexei said, pushing the filter as hard as you can during production, and the storage conditions as well. So this kind of creeped up on us, but when you're performing lab scale filtrations, those headspaces and bottles and vessels, and also subjecting the material to freeze-thaw cycles can inactivate that whole reduction pathway as well. So, I mean, like Alexei said, the smaller scale down preparations of the depth filtration just may also be not observed just due to sample handling and storage conditions of that clarified harvest.
And with that, unfortunately, we don't have time for any more questions. But before I finish, I would like to ask our presenters if you have any closing remarks.
Yeah, so I think this is a good forum, and thank you to Biopharma Asia and all the folks here on the line. This is a pretty interesting topic, and our future work would hope to really kind of look at characteristics of cell culture for integral cell densities, and then also the evaluation of different pore size filters. So with Alexei's data, it was nice to kind of see a similar and then also a positive trend in the evaluation of separate filters that could easily be implemented in future downstream processing.
Yeah, so I would like to thank Biopharma Asia, of course, and all of the listeners, and thank you for great questions. Also, thank you, Brian, for a great presentation. And what I hope, that this elucidates is sort of the need, as we design more and more intensified process, the need for the technology designers and providers to work very closely with the biopharma industry in order to be able to get around some of these issues. Because we can do a lot with our technology, but we need to understand what artifacts are you observing in the present technology, and to be able to work together collaboratively in order to be able to either figure out how to use the present technology in a better way, or how do we design new approaches that are able to get around these issues. So thank you.
So I would like to take this opportunity to thank our presenters, for Brian O'Mara and Alexei Voloshin, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. Thank you, everyone. Have a great day.